Hello friends, welcome to BMH Learning. This video is about allele specific PCR. To detect SNPs, we already have very efficient conventional methods like RFLP, that is, restriction fragment length polymorphism. But these methods rely on the use of site-specific restriction enzymes. But sometimes we have DNA fragments where we can't use a restriction enzyme or the specific restriction site is not found. So how we detect the changes in those molecules? To solve this issue, we have allele specific PCR. So it's a method which is used to detect a single base change, or we can say SNPs, using sequence specific primers. The technique has other names like ARMS, Amplification Refractory Mutation System, PASA, PCR Amplification of Specific Allele, ASPCR for Allele Specific PCR. In most of these acronyms, every time we are using word allele, the reason being is its specific amplification capacity for a specific allele or DNA sequence. So what is ARMS or allele specific PCR? As we know, alleles are alternative forms of a gene. When a mutation occurs, it gives rise to two different alleles for a gene. One type is wild or normal and another one is mutant. The allele specific PCR has the ability to detect this specific allele. So in the example, the wild type has A and T bases, and if we look at the mutant form, it has got G and C. Now the task before us is that whether the given strand is having wild type or it is the mutant one. In order to initiate the ARMS process, we need to design two primers, that is the forward primer and the reverse primer. These primers, as we know, will amplify the DNA. Now, you need to note one thing here, that the site having the SNP is acentric. You can notice it's closer to the one end of DNA and away from other end. Basically, what I'm trying to say is that it's not exactly in the center. You would understand it shortly why we are designing them in such a way. So once the primer designing is done, then we make use of third primer. Now, this primer is designed in such a way that it would bind to the wild type. You can see they are complementary. So T would bind to A. Now let's suppose we are providing all the reagents, necessary conditions, the two basic primers, that is the forward and the reverse primers. So as the amplification process is finished, we are gonna get this, the amplification product. Let's say this is 200 base pairs. Now remember, along with the two primers, we also used a third primer specific for wild type allele. So this primer would also generate an amplified product. But you can notice this one is little bit short. Let's say it's 150 base pairs in length. So we are getting here two fragments actually. The 200 base pair fragment is between these two primers, and 150 base pair fragment is between wild type allele specific primer and the reverse primer. Now let's deal with mutant type. As we know, the DNA of interest in the mutant type is almost similar to the wild type, so we are gonna use the forward and reverse primers of same composition, or we can say the configuration. But regarding the third primer, this is going to be different. This one is specifically designed for mutant allele. So the sequence is different here. Here C would bind to G. So after PCR, As we know, we will be getting two products. The usual product is amplified between these two, and this one is 200 base pair. And about the short one, it's amplified by these two primers, and the length here is only 50 base pairs. Now let me tell you one thing here, that the two specific primers, one for the wild type and another for the mutant one, are amplified simultaneously, and we get these products. Let's remind that the site having the SNP or mutation should not be in the center, but must be closer to one of the ends, that is, near to one of the basic primers. By using this kind of arrangement, we are making sure that the two short fragments, that is 150 base pair for the wild type and 50 base pairs for the mutant type, can be detected or identified in later stages of PCR procedure. This was all. Thanks for watching.